To provide context, efflorescence in ceramics is a serious problem in architecture. It is an indication of material deterioration brought on by salts reaction with ceramic materials. This process occurs when soluble salts, such as sodium sulfate, migrate through the porous material to the ceramic surface and crystallize, often due to ambient moisture. One of the primary chemical types of efflorescence is a mixture of soluble salts, principally carbonates, chlorides, and sulfates. These salts disintegrate and move through the porous structure of ceramics when they absorb water. During this experiment, we looked at the sodium sulfate-induced efflorescence in ceramics. The goal of the study was to comprehend the processes of deterioration that take place when environmental salts are absorbed by ceramic materials, leaving them open to chemical reactions. The materials used in this experiment were limestone rock, ceramic tile or ceramic floor, piece of singular brick, porous concrete with mineral color, sodium sulfate, and 6 plastic containers. And for the procedure, each of the ceramic pieces were kept in a plastic container for the experiment, enabling regulated exposure to the test solution. Then some sodium sulfate solution was poured to the bottom of each container. This chemical was used as a medium to stimulate the ambient conditions that encourage salt absorption and efflorescence in ceramics. After submerging the ceramic specimen in the solution, we snapped pictures to have a baseline against which to compare. Finally, 2 and 24 hours after putting the specimens in the test solution, we went back to the lab to snap more pictures. Now, for the results, there were no impressive results. Only the humidity level of the brick and tabicon increased. We perceive the brick, the tabicon, and the alocreto were affected by humidity in that respective order. Regarding the limestone piece, we can clearly see the effects of crystallized salts. As for the azulejo, there are also signs of deterioration, and finally, in the ceramic floor, there are also no signs of changes. Our analysis showed that after 2 and 24 hours of exposure to sodium sulfate, visible efflorescence developed on ceramic surfaces. This white powdery residue forms when soluble salts migrate through the ceramic's porous structures and crystallize as water evaporates. The degree of efflorescence varied by ceramic type. More porous materials like limestone absorbed salts more effectively, leading to greater crystallization, while denser tiles showed minimal effects. This suggests that higher porosity enhances salt migration and exacerbates efflorescence. We simulated real-world conditions using sodium hydroxide, which raised the solution's pH and accelerated salt dissolution. The implications for architecture are significant. Efflorescence not only affects aesthetics, but can also indicate structural issues, as expanding salt crystals may cause microcracks and eventual failure. In summary, our findings highlight the mechanisms of efflorescence in ceramics, stressing the importance of material choice and environmental factors in design. In conclusion, in this experiment, we now better understand how environmental conditions affect material deterioration in ceramics. We learned how crucial it is to use ceramics with lower porosity when building in areas where moisture exposure is likely. So we can say that we understand the importance of preventing its effects through the implementation of different practices, such as the use of appropriate materials or proper drainage design.